In this video, we'll be seeing a few important terms with respect to DNA. In the previous video, we were seeing the molecular structure of DNA and the different types of DNA with respect to base pairs, the coiling, and the functions they perform. In this section, we'll be seeing about the different terms that is used in common with respect to many DNAs. The first thing we'll be seeing is called as a palindrome. Now, to understand palindrome in a very simple way, I'm going to write a word called level. What is the significance of this word level? Level is spelled as L-E-V-E-L. Now, if I'm going to read it from the backward direction, it is again going to be L-E-V-E-L. So, whether I read it from this direction or whether I read it from the left to right direction, it is going to spell the same. In the same way, if I'm going to say one more word, Malayalam, it is again going to spell the same whether I read from this direction or whether I'm going to read from this direction, whether it is from the left to right or right to left, it is going to spell the same. This kind of a sequence is actually called as a palindrome sequence. So, it is a set or a sequence of nitrogen bases. So, nitrogenous bases include adenine, guanine, thymine, cytosine. It is a set or a sequence of nitrogen bases which will be reading the same in the same direction of opposite strands. That is very important. It will be reading the same in the same direction of opposite strand. What do I mean by that? Now just look at the strands given here, 3' to 5'. It is going to be A, A, T, T, C, G, A, A, and so on. Now again, since I am reading it from 3' to 5' in this direction, in the next strand also, I'll be reading from 3' to 5' direction. And here also it is going to be A, A, T, T, A, A, T, T, and so on. So if you look at the sequence of the nitrogenous bases, here it will be AATTCG, and here also AATTAAT. It's going to be the same. So to keep it very simple, we can tell that the palindrome is going to be the same on both the strands. So whether I read it from this direction or whether I'm going to read from this direction, it is always going to be the same. But what is the significance of this, and where is this palindrome actually very important when you're reading about DNA? Is when you are reading about the replication, the concept of translocation. We'll be seeing it in the further videos. In those areas, this palindrome will be having a significant role to play. So for now, you just need to know that palindrome means it will be reading the same in the same direction on opposite strands. If I read from 3' to 5', it will be reading the same from 3' to 5' on the other strand. Since they are opposite and they are anti-parallel, we should be reading it in the same direction on both the strands. The second concept that we are going to see, the second term with respect to DNA is denaturation. Denaturation means the process by which a double-stranded DNA gets broken or separated into two separate single strand of molecules. You can find it here. So what happens here? There is a double-stranded DNA. This double-stranded DNA gets separated into two single-stranded DNA molecules, and these two single-stranded DNA molecules have come only from this double-stranded DNA molecules, and they are completely separated. Earlier they were together, now they are completely separated. And how is this double-stranded DNA separated? It is separated because of temperature. There is an increase in temperature, and when you increase the temperature, the double-stranded DNA completely gets separated into two single-stranded DNA molecules. Now again, when I am talking about temperature, the temperature is supposed to be specified. There is a specific range of the temperature which has to be maintained for this kind of a breakage of double-stranded or conversion of a double-stranded DNA into a single-stranded DNA to take place. And why is this essential? It is essential because if you keep on increasing the temperature, there is a chance that the phosphodiester bond will completely break. So our purpose is to break only the hydrogen bonds here, because you know that adenine and thymine is bonded with hydrogen bonds. Guanine and cytosine is again bonded only with hydrogen bonds. Our aim is to just break the bond in the middle and to bring two single-stranded molecules. So if you want, you have to eat in such a way where you have to break only the hydrogen bond. If you keep on eating for a long time, then what happens is the phosphodiester bond also breaks. And phosphodiester bond is the main bond which actually links the sugar molecule and the next sugar as well. So there is a single bond, the phosphate, which bonds with the OH on that side, and it bonds with the sugar molecule on the other side also. So if the phosphodiester bond completely breaks, then the whole DNA will get digested. So this would get digested, this would get digested. So the temperature has to be specified in such a way where only the hydrogen bonds are broken and the phosphodiester bonds are not broken. The third term with respect to DNA that we'll be seeing is renaturation. Renaturation is the opposite of denaturation. What is the meaning of denaturation? Nature. What is the natural form of a DNA? The natural form of DNA is double-stranded. I am going away from the nature. I am denaturing it. I am just going reverse of the nature. I am separating it. So that is called as denaturation. I have got a separated DNA. From denatured DNA, I am bringing a normal DNA. So from a double-stranded to single-stranded DNA, I am bringing a single double-stranded DNA. You can find here, there are two single strands separately, and I am bringing a double-stranded DNA here. So when this happens, the process is called as renaturation. And for this process to happen, you need to empower the factors opposite to denaturation. Now see here, for denaturation, what we have done here, we have just increased the temperature. So what is the opposite of it? We need to cool the temperature. When you bring the temperature down, what happens? Renaturation takes place. Two single strands of DNA completely comes together and they form a double-stranded DNA. Now hyperchromatic effect. What is hyperchromatic effect? Now we all know that the purines and the pyrimidines form the part of the nitrogenous basis, and they are the basic keystones in DNA. They are the basic stones in DNA. Now the purines and pyrimidines by nature have a special characteristic feature of absorbing ultraviolet radiation. So by nature, they have got this property of absorbing ultraviolet radiation. Using this property, we can define what is the absorption spectrum of a particular DNA. What do I mean by that? How effective is the DNA or the substances that is present in DNA, how effective they are in absorbing a particular wavelength of ultraviolet radiation. That is what is called a hyperchromatic effect. Chromatic effect means the chrome, this particular, I would call the far length or the spectrum, the particular range in which it is capable of absorption, and hyper, how efficient it is making that. So chromatic means the spectrum, or I can call the range, and hyper means I can call it efficiency. How efficient is the particular DNA molecule is efficient in absorbing this ultraviolet radiation. Now when you do that, what you actually find is that when present in double strand, the purines and pyrimidines, and they are present in double strands, that is, they are present as a double helical DNA strand, they will be absorbing like very less DNA. I'm sorry, they'll be absorbing very less ultraviolet radiation. Why do they absorb very less ultraviolet radiation? Because they are already bonded. Since they are already bonded with the hydrogen bond, because you can find the two strands, they are being bonded here in the central by the hydrogen bond. Since they are bonded completely in the middle, you will not be finding them absorbing much of ultraviolet radiation. So it is very, very less. If you see the amount of ultraviolet radiation that is being absorbed by them is comparatively very, very less. But when they become single strand, the absorption spectrum is very high. They absorb more amount of the ultraviolet radiation when compared to them in double-stranded regions. What is the reason? Because in double-stranded DNA, they are completely bonded with the hydrogen bond. But in a single-stranded, the hydrogen bonds are not there. As a result, the efficiency of that absorption spectrum becomes very higher as well. So these are the four terms which is very important with respect to DNA. The first one is palindrome, which will be reading the same set when read from both the strands in the same direction. The second is denaturation, where a double-stranded DNA molecule is made into a single-stranded DNA molecule. Renaturation, opposite of denaturation, where you bring a double-stranded DNA from a single-stranded molecule of DNA. And hyperchromatic effect, which states that the double-stranded molecules, that is the double-stranded, if purines and pyrimidines are present in double-stranded DNA, the absorption of ultraviolet radiation is very less, and in case of single-stranded DNA, absorption is very high.